So in the past few classes, we discussed how to prepare the cell and then purify the DNA, perform the precision digestion, and perform the ligation, and transform the recombinant DNA molecule into the cell, so that the next step, okay, is to identify a clone of a specific gene that we need to know. So today we will go through this topic. So to identify a clone of a specific gene, then we have this direction selection of a desired gene. So to find the gene, to find the cell, to find a clone that carry the antibiotic resistant gene or marker rescue. These are two common use technique, all right? So like the insertion inactivation, that's what we learned before, okay? Marker rescue, that would be like the autotroph mutant. Those are yeast cells that cannot survive in the absence of specific amino acid, and after the transformation, then you'll be able to get the ability to synthesize a particular amino acid. That's a marker rescue. And also today we will also learn the identification of clone from a gene library, okay, from the technique like nucleic acid hybridization, the colony and plaque hybridization probing, and the immunological detection method. Okay, and then for the direction selection of the desired gene. Okay, so in this case here, so we have the several. So for example, we have this plasmia R65, and then you cut the EcoR5. So you have EcoR5, you cut it, and then you get several fragments, and then you mix with the vector, and then each of these fragments get a chance to be ligated to the vector, all right? And then only one like carry a specific antibiotic resistant gene, all right? That's the one that you want to identify. So from this library, then after the transformation, then you grow the cell in the selective media, and only the vector, okay, only the cell that carry the vector that has the antibiotic resistant gene will be able to form a colony, all right? And that's how you identify this recombinant cell. And the recombinant cell carry the recombinant DNA molecule, okay? And that's the antibiotic resistant gene. And in this case here, and then so you try to identify this the marker rescue, the one that carry the gene that can synthesize the tryptophan, all right? So in this case, they prepare the genomic DNA and cut with a specific enzyme and ligate with the vector, and one of the vector carry the gene that contain the tryptophan, all right? And then you introduce into the bacterial cell, all right? In this case, bacterial cell, and then this cell, because you mix it with the cell, some cell don't get the vector, some cell get the vector, but the vector carry the gene, carry the sequence, doesn't have the tryptophan, and some cell carry the recombinant DNA molecule that has the tryptophan. And then when you grow the cell in the minimum media, which means you don't provide amino acid, and then only those cells that carry the tryptophan will be able to form the colony. So that's called marker rescue. So you have these two different technique. It's called direction selection of desired gene. So I forgot to do the slideshow here. Okay, and then here is to make the genomic library. genomic library so that you can identify a specific colony that you want. Okay, that's the genomic library. So to make the genomic library, so you collect the genomic DNA, and then you perform the precision digestion. So you have a population of the digested DNA, and then those are digested DNA. Then you mix with a population of vector, all right? And the vector, so the vector for cloning vector in an original replication and antibiotic resistant gene and the multiple cloning sites. So you cut with the same enzyme, right, as you cut with the genomic DNA. And then you mix these two populations together, all right? So that this fragment will be able to ligate with the vector. And then after ligation, you introduce into the cell. So each cell will carry one vector which is different from others. All right. So then you have the hundreds, thousands of the colony. And then those colony, each of them will carry one specific piece of DNA. So that's called a genomic library. All right. And also also you can have the differential expression, different gene, right? So that you have cell type A, cell type B, they have the differential expression. And then differential expression, then you can collect the messenger RNA. And from those messenger RNA, then you can make the cDNA. And from those cDNA, all right, you can ligate to the vector and make the cDNA library. Okay, so that's called cDNA library. So that's different between the genomic library and the cDNA library. So these are the how to make the genomic library and cDNA library. And then the probe, okay, as we mentioned about the probe, because to perform the nucleic acid hybridization, the probe basically use the idea of the nucleic acid hybridization. Later on, we're going to talk about the solid hybridization. Okay, basically they use a label to denature the single-stranded probe, and then to hybridize a membrane-bounded DNA, and then the probe must be a label with a radioactive or non-radioactive fluorescence, so that you'll be able to detect a signal. And then the idea is, if the probe complementary to the target, then they will form the stable hybrid. All right. If it's not complementary, then it's not stable. Therefore, this will be washed away, and this will be relevant to later on. We're going to discuss those different detection methods. Okay. So that's the idea of the nucleic acid hybridization. So this hybridization, all right, basically based on the idea of this melting temperature, right? The melting temperature. So as we know that we learned before that the melting temperature, if it's a Tm, right, that means double-stranded DNA, the amount of double-stranded DNA equal to the amount of single-stranded DNA. So in the population, 50% is double-strand, 50% single-strand. At that point, that's the melting temperature. When you increase the temperature, then all the double-stranded DNA would denature into the single-stranded. If you reduce the temperature, then you increase the amount of double-stranded DNA. Okay, so that's melting temperature. The melting temperature is important during this hybridization. Okay, and this is the formula that we learn. All right, to calculate the melting temperature, and for the clone identification, all right, the colony and plaque hybridization probe. So basically, from those, for example, the genomic library or cDNA library, and now you want to identify which specific colony carry the target gene, all right? So what you need to do is you have this plate, and then this plate has many colony. Each of them carry a specific DNA sequence. So what you need to do is you need to use the nitrocellular membrane, all right, to cover the plate so that this cell will attach to the membrane. All right. Once the cell attached to the membrane, they also transfer because each cell carry the genome. So those DNA will also come to the membrane. And then you're going to degrade the cell. All right. You do and also use the alkaline protease. Alkaline basically, so you will denature the DNA. All right. Alkaline condition will denature the DNA. All right, so you denature the DNA, and DNA are exposed on the membrane. And now you treat this membrane with a UV light or 80-degree 2 hours, so that the DNA will intercollect to the membrane, so that will stay. So you fix the DNA on the membrane. And since this DNA has been treated with alkaline, so they are single-stranded, right? They are single-stranded. And then in order to identify the colony, then you have to use the probe, all right? The probe, which is the single-stranded DNA that carry a signal. And then you have to mix with because these are the single-stranded DNA, and then you mix with the probe. Those probe they will perform the hybridization. If it's a perfect match, then they will form a stable hybridization. If it's not perfect, there will be unstable hybridization. Right? So then after you perform this hybridization, and this probe basically is a complementary sequence of the target gene, all right, they will find a specific the colony that carry the gene, the target gene. They will form the hybridization, and after the wash, only the stable hybridization will stay. All right, and then you can expose the signal to the X-ray film. Then you can identify the specific colony that carry the specific gene. All right, so this is so-called colony and plug hybridization, and you use the specific probe and then to hybridize those DNA that attach, that you transfer from the cell to the membrane. Okay, and then this probe, okay, so as I mentioned, it can be a radioactive label or non-radioactive label, because you want to detect the signal. For the radioactive label, all right, so normally we use the alpha P32 ATP, all right, so that means this alpha phosphate is the isotope, so it's a P32. Okay, and then you will have three different techniques to label the probe with the isotope. Number one is called the nick translation. So nick translation, basically, we know the piece of DNA normally sometimes they will have a nick, and then the nick translation, I mean, you add the DNA polymerase 1 plus the P32 deoxy ATP, which is this one, alpha P32 DATP, and then they will form they will synthesize the new DNA strand, because DNA polymerase 1 has a 5' to 3' exonuclease activity, and during this synthesis they will incorporate the alpha P32 DATP into the fragment, and then you get this labeled probe. Or you can do end filling. End filling, that means you treat the DNA with the ECO-1, for example, and then you have a sticky end, and then you use a clinol and the alpha P32 DATP, and then to form this end filling, right? Because clinol will not have the exonuclease activity, so they will only fill in the nucleotide. They will not degrade the DNA. So this is end filling. And another method would be the random priming labeling. So for the the random priming labeling. All right, so you denature your primer, and then you add the random hexamer, oligonucleotide, and then add the clinol and alpha p32 DATP. Then you can synthesize the new strand. In the meantime, you incorporate this p32 DATP into the newly synthesized fragment, so that these three different techniques can be used to label the primer. Okay, and then this primer can be used here to identify the specific coloring that carry the target gene. Any question? Very good. So that's the labeling with the radioactive marker. And this is the example after the radioactive. This is just an example of stolen blood, which we will discuss later. Yes, Hadel.
Yes, Professor.
Yes, Hadel.
Yes.
You have a question?
I said no.
Oh, then you raise your hand.
No, I did not.
Oh, you. Okay, all right. So this is just an example of a stolen hybridization, which we will discuss later. And see, these are the signals from the radioactive label. Okay, and this is an example of a Nolan blot, the Nolan hybridization. Okay, so this after the radioactive label probe hybridized to the membrane, then you see the signal. And then also you have the non-radioactive labeling. Non-radioactive labeling, so we have these two techniques. One is the biotinylation labeling. So basically, you have the biotin label, the DUTP, all right? And then you use the nick translation, M filling, or random priming, and then to incorporate this biotin DUTP. And so that these primer carry the biotin, all right? So when these primer hybridize to the target, all right, then these primer carry the biotin. Then you add the enzyme, all right? So the enzyme carry with the fluorescence marker. So that's the avidin coupled to a fluorescence marker, and then perform the reaction. They will show the signal. And then another one will be the horseradish peroxidase probe. So basically, this single-strand DNA, all right, then you have this horseradish peroxidase and glutaraldehyde. They will attach to the DNA, all right, and then when they hybridize, all right, they carry the signal, and then you add the lumina, and then they give you the chemiluminescence signal. So these are the two non-radioactive labeling techniques, and it doesn't matter whether it's radioactive or non-radioactive. The point is to give you the signal, right? To give you the signal so that you can identify the specific target. So that's the idea, okay? It doesn't matter which radioactive or non-radioactive. And then the next is, and the practical use of the hybridization probing. So this one is abundance probing to analyze cDNA library. So cDNA library. So you have a cDNA library. Then you have a probe A and probe B. Probe A and probe B could be two different genes, so that you can know which gene has been expressed more. All right. So you use two different probe. One is maybe a gene A and gene B, because cDNA library is a collection of messenger RNA that can give you the evaluation of gene expression. So this will give you so in this comparison, you see the probe A give you a low abundance, probe B is high abundance. All right, so you can predict that the probe B, that can be for example, gene B expressed more than the gene A. That's one example. So that's abundancy probing to analyze cDNA library. And so for oligonucleotide probe for gene and the translation product have been characterized. So for example, you have this gene library, and then you have this synthetic and labeled oligonucleotide, and then they identify this specific gene, and then you identify like the clone, and then you can have a predicted oligonucleotide, and then to hybridize the gene library, and then you can have the predicted the function of the predicted nucleotide sequence of a particular clone. And then you have this heterologous probing. So two genes for the same protein but from different organisms. All right, so you have this specific probe, all right, and then they give you this colony here, and then you can use the same probe, all right, and then hybridize with different species, all right, different species, then you can identify it. So you can identify the similar gene, okay, whether it's present in the different species or not. And then from the same species, you want to see a specific function of the gene that can the specific gene of their family. So see how many family carry that specific gene, so you can also perform that the hybridization to the same species gene library clone, and then to see how many appear, so that can give you indication of the similar gene, how many similar gene in that same species. Okay, so that's called heterologous probing. And then for the immunological detection, okay, so immunological detection is just to use the antibody, and then to find the protein in the library. Okay, so basically you prepare the antibody, and then you have the colony, you lyse the cell, and then you add the specific antibody, and to see which cell carry that specific protein. All right, so this is called immunological detection method, to detect the specific protein in the cell. So that's basically to identify a clone of a specific gene from the gene library, or cDNA genomic library, or cDNA library. That's how you identify a clone of a specific gene. And then the next topic will be to study gene location. All right, to study the gene location. So basically the two commonly used method. One is the solvent hybridization, and the other one is the in situ hybridization, and later on develop into the fluorescence in situ hybridization. Okay, these are two commonly used technique, so that you can identify the gene location. Okay. So in previous topic, we identify the colony that carry the gene. And this topic, we're going to study the gene location. So for a solent blot, basically you can use a solent blot to determine the gene copy number, gene mapping, gene mutation detection, PCR product confirmation, and DNA fingerprinting. That's the solent blot. And basically the technique for the solent hybridization is you collect the genomic DNA, and then you perform the restriction digestion. Okay, after the restriction digestion, you perform the gel electrophoresis, so that you will see a series of the DNA fragment, different size of DNA fragment, because these are fragments after the restriction digestion. And after the digestion, then you transfer these sequence, all right, from from this electrophoresis to a membrane. And later on, you use the probe and to identify the presence of a specific fragment, and also determine the size of the fragment, and so that you'll be able to determine the location. Okay, so this is more specific detail about the technique. So you collect the genomic DNA, perform the restriction digestion, and then you perform the gel electrophoresis. So after gel electrophoresis, you should see a smear on the gel, and then you transfer to the nitrocellulose membrane. All right, nitrocellulose membrane, and then you fix the DNA on the membrane, and then you hybridize with a specific probe and wash, and then you get the signal. All right, and this is even more detailed protocol about the southern blot hybridization here. So you have let's say you have these different preparation, all right, and each preparation you use the so first preparation is the size marker, and second preparation you perform the digestion of one particular enzyme, and then this is another particular enzyme, and then you all right, and digestion, and then you perform the gel electrophoresis. So the first one is the size marker and number 1 enzyme, number 2 enzyme, and then transfer to the membrane. All right, and then membrane you perform the hybridization. Then you have a size marker, and then 1 enzyme, second enzyme. And then from this information, then you can interpret the the specific gene location. All right, and then in this part of transfer, all right, so we have 2 different type of transfer. One is called capillary transfer. So the capillary transfer, so that means you have the salt buffer in the bottom, and then you have this membrane paper that can draw the buffer to here, and then you put the agar gel, and then the membrane on top of this, your paper towel plus a heavy object, so that this buffer will be absorbed and go upward, and that's called capillary transfer. During this process, the DNA will be transferred from the gel to the membrane. Okay, so these are all the setup for the capillary transfer. All right, this is capillary transfer. Okay, and then another technique would be this vacuum transfer. So basically the vacuum transfer, so you put a gel on the top, you have the filter in the bottom, and then you have the support here, and the porous plate, and then you have a buffer here. And then you use a vacuum so that to draw the buffer down. During this process, the DNA will be transferred from the gel to the membrane. Okay, so that's the southern transfer. Any questions so far? Okay, very good. So now we're going to watch this because the southern transfer is very important. So I want you to watch this video carefully. Okay.
These 4 cells represent different clinical isolates of the same species of bacteria. Each contains plasmid DNA, but the plasmids in one isolate may not be the same as the plasmids in another isolate. We can purify the plasmid DNA from the isolates and use 2 procedures, gel electrophoresis and southern blotting, to compare these plasmids. As a first step, we can use gel electrophoresis to examine the plasmids in their native form. Blue loading buffer is added to an aliquot of the plasmid sample, and the mixture is loaded into wells of a jello-like slab made of a polysaccharide material called agarose. The agarose contains microscopic pores through which the DNA can travel. An electric current draws the negatively charged DNA through the gel toward the positive electrode. The positions of the DNA on the agarose gel indicate the sizes of the plasmids. Smaller pieces of DNA migrate faster than larger ones through the agarose pores, because larger pieces meet with more resistance. In this example, the plasmids in lanes A through D are similar in size. The plasmid in lane C migrates slower than the others and is therefore a little larger. Although the plasmids in A, B, and D appear to be the same, we must perform other tests before we can come to this conclusion. Our next step is to add enzymes called restriction enzymes to the plasmids. An example of a restriction enzyme is EcoR1. A restriction enzyme cuts the DNA at a particular recognition sequence. EcoR1 recognizes and cuts the sequence GAATTC. If the plasmids in the different tubes are identical in sequence, they should have the same number of EcoR1 recognition sequences. The type of plasmid in this particular sample can be cut in 4 places with EcoR1, producing 4 fragments of different sizes, represented here as different colors. When the fragments are loaded on a gel and electrophoresed, the fragments separate from each other. The slower bands on the gel contain larger fragments, and the faster bands contain smaller fragments. The patterns of fragments in lanes A and B appear identical. On the other hand, the patterns of fragments in lanes C and D appear unique. Are the DNA sequences and fragments in lanes A and B the same or nearly so? And is there any similarity between them and any of the fragments in lanes C and D? To answer this question, we perform what is called a southern blot. An apparatus is set up to transfer the DNA in the gel onto a solid support, such as nitrocellulose paper. The gel is placed on top of a sponge, soaking in an alkaline solution. Nitrocellulose paper is placed on top of the gel, and a stack of paper towels is added on top of the nitrocellulose. The solution is drawn to the stack. The DNA denatures in the alkaline solution as it's carried up the stack. The DNA stops traveling when it reaches the nitrocellulose. If we could see the DNA on the nitrocellulose paper, it would show the same pattern of DNA bands as existed on the agarose gel. We place the nitrocellulose paper into a plastic bag and add a solution containing a radioactive single-stranded DNA called a probe. In this case, the probe consists of all the fragments from plasmid A. During an incubation period, the probe anneals to complementary sequences, forming base pairs with the DNA in some of the bands on the paper. The excess probe is washed off. The only probe that remains on the nitrocellulose paper is the probe that is annealed to complementary DNA on the paper. After the probe is washed off, the paper is overlaid with X-ray film. The film and nitrocellulose are kept in the dark for a period of time, and then the researcher develops the film. The radioactivity in the probe creates black bands on the X-ray film, which is called an autoradiograph. The autoradiograph provides more evidence that plasmids A and B are the same or very similar. However, plasmid D is unrelated to plasmid A, from which the radioactive probe was derived. Plasmid C has some similarity to plasmid A, but the similar sequences are found on different sized DNA fragments. The autoradiograph gives us more information than the agarose gel on its own.
So that's the southern blot hybridization detail. Okay, it's a very important technique. Okay, so now the next technique to study the gene location is called in situ hybridization. All right. So in situ hybridization, basically you prepare the cell on the slide, and then you break the cell and fix the cell, and then you use the probe, and then to hybridize the cell, and then so that you will see the location of a specific the target gene on the cell. Okay, that's called the in situ hybridization. And if you use a probe, that the probe is labeled with the fluorescence antibody, then we call the fluorescence in situ hybridization. It's called FISH. All right. So the for the FISH, that can perform a direct examination of the chromosomal DNA molecule. Okay. In this technique, a cloned DNA fragment is labeled. All right. So we have the DNA the DNA fragment is labeled with a fluorescence marker, and then we hybridize to the preparation of a chromosome that has been immobilized on a glass slide. Okay, and then the physical position of the DNA fragment within the chromosome can then be revealed simply by examining the preparation with the microscope, and then you can see that in more detail from the computer. So basically, so then you can see this FISH technique involve the hybridization of these labeled DNA probe in situ a chromosomal target, right? And the standard FISH probe protocol has 5 steps. The first step is sample pre-treatment, and then denaturation of probe and hide sample. And number 3, hybridization of probe to the target cell or metaphase spreading. Number 4, post-hybridization washing. And finally, detection using a simple fluorescence microscope with the appropriate filter so that you can be able to see it. So that's the FISH. And for the FISH, you can do the metaphase FISH sample. So basically, for the metaphase, then you prepare the cell, like you culture the cell for 72 hours. Then about 4 minutes before the harvesting, you add the chemicals called cosmic, and the cosmic will arrest all the cell in metaphase. All right, and then the cell can be suspended in hypotonic media, and then fixed with the methanol acetic acid. And then these fixed cell suspension you can apply to the slide and let it dry, and then the target DNA in the cell is fixed and denatured on the surface of slide, and then you can add a probe DNA to detect the presence of your target. Okay, that's a FISH. And here is just a general procedure to perform the FISH. And so you can use a FISH to analyze genetic disorder, or you can also determine the expression of a particular messenger RNA. Okay. And so I previously introduced the metaphase FISH, and also you can do the interphase FISH. And to prepare the interphase FISH, basically, it's very important that you have to permeabilize the cell for optimum probe-target interaction and maintain the cell morphology. Okay. So basically, you have to incubate the cell overnight after deposition on slide. And then after aging overnight, cells are treated with the protease to minimize interference from the cytoplasmic protein, and fixed with 1% of formaldehyde to stabilize the nuclear morphology. And before the DNA dehydration, the cells are dehydrated in a greater concentration of ethanol. All right. So that's how you prepare the interface and the fish. And because interface fish does not need to culture the cell and stimulate the division, it is valuable for analysis of cell to not divide well in the culture. And metaphase is useful in detecting small or complex rearrangement that are not by regular chromosome bending, like G bending, okay? And then the fish probe. So the probe that you use to identify the specific targets, like this, you can see these are the small bright white spot. Those are the indication of the hybridization from probe to the target. And the probe can be a whole chromosome painting probe to decipher the cytogenetic aberration, or repetitive sequence probe for chromosome enumeration, that including the centromere enumeration probe, or locus specific identifier LSI probe for gene fusion, gene deletion, or duplication. And the WCP basically designed to mark the entire chromosome of interest. And it's all right. So like this is whole chromosome painting, like this in green. This is chromosome number 6, all right. And this is also labeling sperm, the nuclei with WCP probe, right, to analyze the sperm chromosome segregation. So here the chromosome number 13 painting probe, you label as green, all right, the label is green, and also a chromosome 22 painting probe label in orange. Okay. And this is another example of interface fish, and use the centromere enumeration probe. Okay. So they can hybridize to the alpha satellite repeat sequence in the centromere, so that you can perform the chromosomal enumeration. And here they can detect the CEP, the centromere enumeration probe, detect the aneuploidy of chromosome. And here they show the XXY hybridization pattern. And here is also this is the locus specific identifier probe, so they can identify the aneuploidy. So here you see these, and then here you have 3 is a disomy, all right, and this is disomy, chromosome 2, and this is trisomy, all right. This is trisomy. You have 3 signal here: trisomy 2, disomy 2. And here you can use the interface dual color fish to detect the gene rearrangement. And also you can use a telomere probe to determine the chromosomal structural abnormalities. All right, so these can see the telomere. And then the advance in fish technology. So basically, has been developed several molecular cytogenetic technology. Okay, so like the comparative genomic hybridization, the CGH, the spectral karyotyping, that's SKY, multicolor fish, that's MFISH, multicolor bending, MCB, and also array CGH. So these are used for to determine the gene location and also the gene mutation. And this is just the example of the multicolor fish, all right? So different chromosome and give you different color, okay? And the multicolor banding from the same chromosome, they give you a different banding, so that you'll be able to detect the mutation, okay? And because different banding, they give you the location of specific gene, so that you will be able to determine whether the change of the location of the specific gene. And then the comparative genomic hybridization, CGH, is a very efficient approach to the genome-wide screening, right, for the chromosomal copy number change. So basically, the chromosomal CGH based on the quantitative two-color fish, all right? And so basically, you have two color, and then you compare the signal at the specific location. Then you can determine that specific gene whether it has been upregulated or downregulated. Okay, so it's just like this. So you have reference, and then you can see whether it's been deleted. All right, so you show the different signal, so you can compare been amplified or been deleted based on the color. So that's the comparative genomic hybridization. So basically, you have the labeled DNA hybridized to the to those the normal chromosome on a microscope. Then you can compare to the difference between the normal and reference. And this is example that so after the hybridization, they can see this different signal, and then to tell you whether the area of the gene has been amplified or been deleted. And this is just a typical comparative genomic hybridization. So you have a mixture of a test DNA from a patient and also from a normal reference, and then they are labeled with different fluorescence and hybridized to the normal chromosome spread. All right, this is normal chromosome spread, and then after the hybridization to the chromosome 13, and then you'll be able to compare the signal difference, and then to determine the gene has been amplified or deleted from the chromosome. And then, although the chromosomal CGH increase the potential for identifying new chromosomal abnormalities, and that technique is time-consuming and also does not significantly improve the resolution. All right. So then later on, they develop this array CGH. Okay, array CGH. Basically, they use a technique like this. So basically, you have this array, right? Arrays, and then this array, each spot carry a specific DNA probe, and that probe will come from a specific location of the gene in the chromosome. And then you have this labeled reference DNA and labeled sample DNA mixed together, and then use this mixture to hybridize this array. And because we have hundreds, thousands of different probes on this array, each one represents a specific gene from the chromosome. All right, and then from this signal, based on the hybridization result, then you will be able to make a map like this, because this whole chromosome map based on this probe. Okay, each probe will define a specific location on the chromosome, and then based on the hybridization result, they give you this signal so that you can determine which gene has been amplified, which gene has been deleted. So this will be a more accurate and also a higher resolution. So that's called array CGH. Okay, So it's kind of a combination of the fish and also the microarray. All right, and then you'll be able to determine the specific genes amplification and the gene deletion. So that's the end of today's lecture. Any question?